Genetic engineering is a process which involves editing the genome of an organism. In genetic engineering, the gene that gives a desirable characteristic in one organism is cut out and inserted into the genome of another organism. In this way, the organism which has been genetically modified will gain a new desirable characteristic. This is useful if a disease is drastically affecting some plants. A plant could be genetically modified in order to gain resistance against the disease. Bacterial cells can be genetically modified in order to produce useful substances. For instance, they can be modified to produce insulin. Animals can also be modified in certain ways. However, it is in plants that scientists have seen the most progress. GM crops are crops that have been genetically modified. Crops can be modified to produce higher yields and to reduce the impact of pests. Specific examples include potatoes being modified to be more resistant against pests, and crops being made resistant against mosaic viruses. Concerns about genetic engineering include the unknown long-term effects it could have on animals and plants. For instance, if humans eat GM crops, there could be unknown health risks associated with them. Another issue could be the spreading of these genes. If a desired gene is given to a plant population, the gene could pass on to the wild, unmodified organisms and cause some serious issues. For instance, if a certain gene which gives a plant resistance to pesticides is passed on to a pest, they could become resistant too. Despite this, genetic engineering could have some really good uses in medicine. As we have already seen, there are some ways in which to produce large volumes of insulin using genetic engineering. Also, genetic engineering could be the answer to overcoming genetic disorders. GM crops also have some pretty good advantages, such as being able to survive droughts or floods, being designed to survive in unfavourable conditions, such as in dry climates, and all these lead to a higher yield of crops compared to unmodified plants. This next section is higher tier content only. The steps involved in genetic engineering are as follows. Enzymes are used to isolate and then cut out the gene which gives the desirable characteristic from the organism's DNA. The gene is inserted into a vector using different enzymes. A vector is used to insert the gene into the cells of an organism. A common vector is the plasmid found in bacteria, but viruses can also be used as vectors. The cells containing the new gene will divide, and then as the organism grows from a young age, the characteristic will be displayed. When an individual has been cloned, it means that another organism has been formed by asexual reproduction, and so this organism is genetically identical to the parent. Cloning usually takes place if an animal or plant shows certain characteristics, and these characteristics don't want to be lost. If you wanted to clone a plant, it can be done quite easily. All you need to do is take a cutting of the plant, dip it in some growth powder, and plant it. The plant that will grow will have the same genome as the original parent plant. However, there are some more effective ways than this. Tissue culture allows many cloned plants to be grown at once. A sample of the tissue from the parent plant is taken. These tissue samples are grown in a hormone mixture to stimulate the development of identical plant tissues. The mixture is then changed so that it is suited to encourage the development of the plant. If this is done with many samples at once, then many identical plants are also formed. As you might expect, cloning animals is most common in farming. An animal with certain features, such as producing high quality milk, can be cloned so that the genes that give this feature are not lost. Embryo cloning is used for this process. An animal is given hormones to produce many eggs at once. These eggs are then fertilised either by collecting the eggs and doing it in a lab, or it is done inside the animal. If fertilisation takes place inside the animal, the fertilised eggs are collected afterwards. The embryos are then split, and each embryo cell is put into a surrogate mother. The embryos will then develop into identical offspring. This method is often used on cows. There are some worries about this method, as people are concerned, again with the gene pool decreasing, and the controversy around actually carrying out this method. Some people question the ethics of this process, as it is not natural and the surrogate mothers have to take hormones in order to prepare them for pregnancy. This embryo cell cloning is not strictly a true clone of the parent, as sexual reproduction is still used. So the offspring are identical to each other, but not to the mother or father. Adult cell cloning is a method which gives clones of an animal without using sexual reproduction. In order to do this, a nucleus is taken from a body cell of one animal. Also, an egg cell of another individual is taken and the nucleus is removed. The nucleus of the first animal is placed inside the now empty egg cell. A small electric shock initiates the division of the cell into embryonic cells. Once these cells have started dividing, the embryo is put back into the womb of a female of the same species. The offspring will be identical to the individual from which the nucleus was taken. Adult cell cloning could save animals from extinction, and it could also have future medical applications. However, once again, there is a worry about the gene pool if this method is used. People are also worried about this method as it could be abused in farming or even in producing cloned human babies.